In the following video, we're going to be talking about transcription and translation.
First, we're going to talk about transcription from DNA to RNA. In the first step of transcription, DNA is unwounded by RNA polymerase. One of the strands is labeled as antisense strand and the other one as a sense strand. One goes in the 5' to 3' direction, while the other one goes on the opposite direction.
In step 2 of transcription, an enzyme called RNA polymerase constructs the mRNA by replicating the DNA antisense strand in a 5' to 3' direction. RNA polymerase uses complementary base pairing in order to create mRNA. It is important to keep in mind that in RNA, thymine is replaced with uracil, and the base pairs are guanine with cytosine and adenine with uracil. In step 3 of transcription, we can see how the mRNA strand has been created. We can see that it has different bases, but 3 of them are circled. This is done because these 3 bases make up a codon. It is important to know that any 3 bases in an mRNA strand make up a codon. It is also very important to know this because chains of codons make up amino acids. In the 4th step of transcription, we can see how the antisense strand is labeled with initiator and terminator. This is because the RNA polymerase starts synthesizing mRNA from the initiator and ends when it reaches the terminator. That is when the process of elongation of mRNA ends. Step 5 is the last step of transcription. In this step, the RNA polymerase reaches the terminator and the process of creation of the mRNA transcript is terminated. Once this happens, the mRNA transcript is released from the antisense strand as well as the RNA polymerase. In these 5 steps, we have been able to see how the process of transcription from DNA to mRNA works. Now, we're going to be discussing the process of translation.
In step 1 of translation, the antistrand is transcribed in the sense strand in order to then be translated into protein. Step 2. The ribosome binds to an mRNA at a specific area. Step 3. The ribosome starts matching tRNA anticodon sequence to the mRNA codon sequence. Finally, step 4. The ribosome continues until it hits a stop sequence. Then it releases the polypeptide and the mRNA.